Electrical phenomena of cells, meaning this idea that cells exist in a polarized state. They are not, there's not an equal charge on both the inside and outside of the cell to make it neutral. They exist in this polarized state that allows them to become stimulated when there is a sufficient stimulus available. And the reason that they exist in this polarized state of unequal charges, and what contributes to that is the relative concentrations of sodium and potassium mainly. There are other solutes and there are other molecules and compounds at play, but primarily we talk about sodium and potassium. Sodium is primarily outside the cell, potassium is primarily within the cell. So we call sodium the primary extracellular electrolyte. We call potassium the primary intracellular electrolyte. And we'll see how these will move in order to produce um, changes within a cell. So here you can see we are polarized. We have a difference in charge from the inside to the outside. Lots of sodium on the outside, very little on the inside. Potassium, lots on the inside, very little on the outside. Now, the only thing I don't like about this picture here is it shows sodium only on the outside and none on the inside, and it shows potassium only on the inside and none on the outside. This is not exactly how our cells exist within our body. There is some sodium within the cell, and there is some potassium outside the cell. It's just in a greater concentration, either inside or outside. So that's what this picture is going for, to tell you, hey, there's mostly sodium is outside and mostly potassium is inside, but understand that there are really both on both sides of the cell, intracellularly and extracellularly. Now, if sodium has a +1 charge and potassium has a +1 charge, how are we getting this polarity? How are we more negative on the inside? What are all these negative other charges located within the cell? Well, there has to be something in there that's contributing to this negative charge. And what we see is lots of proteins, some of our nucleic acids and other compounds within the cell actually have a net negative charge. And so they contribute to the negative charge on the inside of the cell and contribute to our cell's polarity. So contribute to them being polarized. Okay. So you can see here we are polarized. When we talk about muscle physiology and the nervous system, we will talk in more detail about the specifics of the polarization on the inside relative to the outside. So we have this electrical potential, this resting potential, with the charge being negative inside relative to the outside. Okay, so you talk about it relative to the inside of the cell. The inside is more negative than the outside. Okay, but when we talk about how muscles contract and we talk about action potentials in the nervous system, there's a movement of ions. What you have is you have some of this sodium coming in, you have some of this potassium going out. And so if we do that, if we change the concentrations, if now we bring some sodium in and bring potassium out, now we're disrupting this polarization. So what you have to do is you have to restore the relative concentrations of sodium and potassium. And the way you do that is through the sodium potassium pump, or the sodium potassium ATPase pump that we alluded to a little while ago. The fact that it is requiring energy, ATP, and so it is primary active transport. It pumps 3 sodium molecules out of the cell and brings 2 potassium inside of the cell. So let's answer some questions here. What type of cell junction allows for communication between adjacent cells? Hopefully you recognize this as being a gap junction. Passive movement across the cell with a carrier protein is termed what type of transport? This is going to be called facilitated diffusion. It's still diffusion, it's still passive, no energy, but now we require a protein. True or false? Phagocytosis is a type of exocytosis. Hopefully you recognize that as being false. Phagocytosis is a type of endocytosis, which is you're bringing something in from the extracellular space. And then lastly, true or false? Cells are relatively equal in charge inside relative to the outside. That is also false. It's what we just talked about. Our cells exist in a polarized state. We are more negative on the inside relative to the outside because of our uh, different negatively charged proteins and polymers and other things like nucleic acids within our cell. So let's now talk about our cell, and we'll talk specifically about the various organelles. So we talked about earlier that we have the cytoplasm, and the cytoplasm is really two things. It's the cytosol, which is the liquidy part or aqueous component, and then we have the organelle, which you'll call a particulate component. So particulate meaning a particle, something physical there. So we'll take a look at these different ones. Now, the mitochondria. This is going to be our first organelle. It's relatively small and rod-shaped. This is where cellular respiration occurs. So this process of taking generally you think of a glucose molecule, breaking that down into smaller components, giving off carbons as you go in the form of carbon dioxide, and ultimately you make energy. Okay, you talk about this more specifically later in another course, but you can think of it as being the powerhouse of the cell. So you hear powerhouse of the cell all the way from middle school to high school to undergraduate to graduate level education. Now, the thing that changes is the meaning behind powerhouse of the cell. In middle school, all it meant was it makes energy, and that's fine. In high school, you add a little bit. Undergraduate, you add a little bit more. In graduate school, you add a little bit more. So the emphasis you place behind it changes, but the meaning of it really doesn't. It's still the powerhouse. It's still where we're making our energy as ATP. And what you may have noticed by looking at it is it is a double membrane. It contains a double membrane. It contains this outer one, which you'll call the outer membrane, and then this inner one called the inner membrane. These invaginations that you see of the inner membrane are termed cristae or crista singular, and there is a space in between this outer membrane and this inner membrane known as the inner membrane space or intermembrane space. And there are some important processes that occur there when you talk about cellular respiration and oxidative phosphorylation. Now, our ribosomes are our next organelle. These are small, round, and granular-like. They can either be bound or free, meaning they can be attached to something or free-floating in the cytosol, and they're responsible for the assembly of our amino acids into proteins. So we'll talk specifically about this with our protein synthesis lecture of how we bring the monomers of proteins, which are amino acids, together to make a polypeptide chain, and then that begins to twist into a more three-dimensional protein structure. We have endoplasmic reticulum. Endoplasmic reticulum is this double membrane coiled organelle. It exists in two forms, smooth and rough. What makes one smooth and what makes one rough is the presence or absence of ribosomes. So we set up here that they can be bound. If they are bound, they are that makes it a rough ER. If a smooth ER does not have any ribosomes bound, or if an ER has no ribosomes bound, it is smooth. Okay. Smooth ER is responsible for synthesis of lipids and different hormones. It stores calcium in muscle cells. It produces phospholipids, detoxifies toxins. Now, in muscle cells, you don't call it a smooth ER, a smooth endoplasmic reticulum. You call it a sarcoplasmic reticulum. But what it does is it serves as a storage location for calcium, and then that calcium will ultimately be released during the muscle contraction process to help initiate the actual muscle contraction. Now, rough ER, it is covered with ribosomes, so it's called rough, and is largely involved with the synthesis of proteins. And so we'll look at that with the protein synthesis lecture. So here's a picture. Here you can see smooth ER over here. Smooth, you don't see any ribosomes. Here is rough ER. This is studded with ribosomes all over its surface. So our next one is the Golgi, sometimes called the Golgi body or the Golgi apparatus. It's flattened and sac-like. It looks something like the ER, but slightly different. And you can think of this as the post office of the cell. It's going to receive products. It's going to package them. It's going to sort them into different regions. And then it will ultimately ship them off in membrane-bound vesicles like we talked about earlier. So the vesicle will be it will be surrounded by a membrane from the Golgi. It will then travel. It'll fuse with the plasma membrane if it needs to go outside of the cell, or it may be packaged to go to a different part of our own cell. Okay. Our lysosome. We alluded to a lysosome previously in the lecture. This is a small organelle sac-like structure that contains digestive enzymes. And these enzymes can be used to break down old cell parts, dead cell parts, things that are not necessary anymore, or if we phagocytize and we bring something in, this lysosome can bind with that phagosome to form a phagolysosome, and then those digestive enzymes can begin to break it down into more manageable material that can then be expelled out of the cell via exocytosis. All right, so here's a Golgi. You can see it somewhat resembles a, a, um, a rough ER. It's not located as close to the nucleus. It also doesn't have any ribosomes on its surface. And usually in depictions, in artist depictions, they will draw in these little free circles. And what these are your membrane-bound vesicles that we just alluded to. So they products will come in from one side, be packaged, and then exit on the other side in these membrane-bound vesicles. All right, peroxisomes is our next one. They're like lysosomes, but they break down substances via H2O2. So H2O2 you may recognize, you may not. It is hydrogen peroxide, and these are common in the liver. They're common in the kidney. These peroxisomes are largely responsible for lots of different detoxification, but specifically alcohol. So you're able to change the number of peroxisomes you have based on how much alcohol you consistently consume. If you consume more, you generate more peroxisomes. You're able then to detoxify and metabolize alcohol more quickly. And so this is what you can think of as building up a greater tolerance to alcohol. Whereas if you did that for a while and then you stopped and you didn't drink any alcohol for a long time, that tolerance would begin to go away. The peroxisomes begin to go away, and you're not able to drink as much alcohol at a given time. Now, our cytoskeleton is our next one. What these are is a series of um, filaments and proteins that serve to support the cell, maintain its integrity, as well as participate in some cellular functions like cell division or mitosis. So you have microtubules, microfilaments, and intermediate filaments. This intermediate filaments really should be in the middle because it's the mid-size one. Microtubules are the largest of the three, and then microfilaments are the smallest. These are all made of various protein structures that serve to, like I said, keep the cell supported, maintain its rigidity and integrity, as well as help with a little bit of cell movement. So our last one, I believe, here that we're going to talk about, there's actually a few more, is the centrioles. And so this is centrioles. This is one centriole. This is one centriole. A pair of centrioles here is termed a centrosome. And it's out of this centrosome that we produce microtubules during cell division. They will radiate out. They will connect to the kinetochore of our chromosomes as they align at the metaphase plate. And then these microtubules will begin to essentially reel these chromosomes or sister chromatids apart and pull them to their respective sides of the cell. And so if you look closely at these centrioles, sometimes they're described as having a 9+3 arrangement. And so all that means is you have 9 groups of these proteins here. You have 9 groups of these, and these are made of microtubules as well. 9 groups, and each one contains 3. So if you count all these, there's 1, 2, 3, 4, 5, 6, 7, 8, and 9. And each of those 9 contains 3 microtubule parts. So it's a 9+3 arrangement of the centrioles. And again, a pair of centrioles is referred to as a centrosome. So here's just an overview of our eukaryotic cell, the different organelles that you can find within it. you should be able to recognize most of them. Golgi with their vesicles, rough ER because you see that they're studded. This is smooth ER. You can see mitochondria, centrosome, so on and so forth. So our nucleus, if we go back, you can see a large nucleus here. Our nucleus is going to be our largest organelle. Some may have multiple nuclei. Some will only have a single one. Um, our skeletal muscles sometimes have more than one nuclei, whereas other cells, general epithelial cells, smooth muscle, um, uh, neurons, typically just have one single nucleus. Okay. Sometimes the nucleus can be helpful in determining what cell you're looking at. So varying types of leukocytes and white blood cells, I think I talked about this at the very beginning, you can sometimes differentiate them based on their nucleus and based on the appearance and location of their nucleus. It will give you clues as to what cell you're looking at. Now, surrounding the nucleus, just like many other organelles in our cell, we have a membrane, which is called the nuclear envelope. And you'll see throughout that nuclear envelope there are these little perforations. There's these little holes called nuclear pores. And what these nuclear pores serve to do and serve to allow for is things coming into and out of the nucleus. Well, you may ask, what needs to get in the nucleus and what needs to get out of the nucleus? Well, in protein synthesis, for example, when we talk about it in our lecture, we'll see that we make something called an mRNA strand, and we make that within the nucleus. But the next step that uses that mRNA strand occurs outside of the nucleus, in the cytosol, in the cytoplasm. So you have to get that out, and the way it leaves is through one of these nuclear pores. So you have your nucleus, you have your nuclear membrane surrounding it, and you have these little perforations all throughout that are your nucleopores. Now, the nucleoplasm is analogous to the cytoplasm, essentially, or the cytosol within the cell itself. It is the fluid that within the nucleus that contains our genetic material, our DNA. So all of this twisty material that you can kind of see blurred out and grayed out behind this blue structure is all of our genetic material. It's in the form of chromatin right now, meaning it's uncondensed. And whenever it begins to condense and form into actual chromosomes, then this chromatin becomes chromosomes, and that occurs during particular stages of our cell cycle. So within our nucleus, we also have a smaller organelle called a nucleolus. And within this nucleolus, this is where we begin to create and synthesize and manufacture our ribosomes. And those ribosomes will then be put out into the cytosol, where they can either be free or bound, like we talked about earlier. So here you see the chromatin existing in its uncondensed loose form before it becomes condensed chromosomes, and then you can see a nucleolus right here. So we'll answer these quick questions real quick. True or false, the Golgi apparatus exists in a rough and smooth form? You should answer this as being false. It exists in a single form. The endoplasmic reticulum exists in a rough and smooth form. What serves as the site of cellular respiration? Which organelle? This is the mitochondria. Where are ribosomes produced? This is what we just talked about. This is ribosomes are produced in the nucleolus, which is within the nucleus of the cell. And then what three structures contribute to the cellular cytoskeleton? We know we have microtubules, you have intermediate filaments, and you have microfilaments. So we will stop here and we'll pick up with